Everyone, in this video I'm going to briefly talk about what makes a cell. Then I'm going to talk about some of the big differences between prokaryotic and eukaryotic cells, and then I'll wrap up talking about how we think eukaryotic cells may have originated. So what is it that makes a cell a cell? Well, it's actually pretty simple. Cells share two essential features. They're surrounded by a cell membrane, and they contain genetic material. So in other words, they contain the key information that's passed on from a mother cell to a daughter cell. And once again, to bring this definition to life, it can be helpful to contrast these traits with the traits of viruses. So viruses also contain genetic material. However, in their case, the genetic material is actually surrounded by a protein coat called a capsid. And sometimes this capsid's also surrounded by an outer layer of lipids. That's shown here as this coat or viral envelope. So this outer layer of phospholipids, but not always. So again, it's just these two basic features that make cells cells. And for prokaryotic cells, things don't really get that much fancier than a membrane and some genetic material. And the little cartoon I'm showing you here shows you the major moving parts of that generic prokaryotic cell. So the hereditary information for the prokaryotic cell is its DNA. So that's this purple noodly stuff here. Then the liquidy substance that's found inside of a prokaryote is referred to as its cytoplasm. So the word cytoplasm, the prefix cyto means cell, and the suffix plasm means liquidy substance. And then also pictured in this cartoon, these little orange specks inside of this cell are protein RNA clusters known as ribosomes. And there are certain types of prokaryotes that also have the other specialized structures that are illustrated here. So the pilus or pili are these long, thin projections of the cell membrane that are a way for a prokaryote to interact with its surroundings. And then there's some prokaryotes that also have this long, whip-like tail called a flagellum, and that can be used to move around in the environment. And then there are other types of prokaryotes that have some additional bonus layers of protection outside of the cell or the plasma membrane. So here's our plasma membrane in this pale orangey color. And so those outer layers are the prokaryotic cell wall, shown here, and then outside of that a capsule layer. And the prokaryotic cell wall is built out of peptidoglycan. And so as the name implies, peptidoglycan is a polymer that's made out of a combination of sugars and amino acids. So glycan, like glycogen, peptido is like a peptide bond, so it's the amino acid part. And this cell wall helps to create some rigid structure so the prokaryote doesn't wind up being smushed flat all of the time. And then this outermost capsule layer is usually made out of polysaccharides, and this can help to keep a prokaryote from drying out, and it can also help it to stick to different surfaces and stay in place. So again, that cell wall and the capsule are present for some types of prokaryotes, but not all of the prokaryotes. So now for jumping from prokaryotes to eukaryotes, we've got a couple of basic observations and distinctions to consider. The first is that prokaryotes tend to be a lot smaller than eukaryotes. So here the average prokaryote is somewhere between 0.1 and 5 micrometers in diameter, while a generic eukaryote can be somewhere between 1 to 100 micrometers in diameter. I find it can often be hard to grasp what these size differences mean, so we're going to take a second to check out a website that lets us zoom through things to get a sense of size. Okay, so here's our website. It is at htwins.net/scale2 if you want to check it out yourself. And right here we've started out at a 1 meter circle. We're just going to start zooming in. So here's relative sizes of a hailstone, a basketball, a ruler, and a teapot. So if we start to zoom in, some other familiar objects, so hummingbird, matchstick, earthworm, shrew, some eggs and a penny. There's the wavelength of a microwave. Coffee beans, coffee. Marble, a sunflower seed. Now we see an ant appear. There are some ants bigger than this. There's duckweed. And now we've reached a scale here at 0.001 meters. So that's 1/10 of a centimeter, where you can see the largest bacteria and also the size of things like a grain of salt or a grain of sand, and dust mites and an amoeba. So here we've got a mixture of prokaryotes and actually large eukaryotic, or tiny eukaryotic organisms. The amoeba is also a eukaryote. Zoom in further. We've got a paramecium, a human egg, smallest things that we can see. And as we keep going in, here's some funny size units. Notice we're now at 10 to the negative 4.3 power. And we're seeing mist droplets, skin cells. And as we zoom in further, here's the size of a red blood cell and some structures inside of cells, the chloroplast and the mitochondria. And here's an E. coli, so one type of prokaryotic bacteria. We keep going in. Now we're going to encounter viruses. And then the smallest thing you can see on a microscope. And even smaller viruses, so the HIV virus, hepatitis B virus, porcine circovirus. And now we're at a molecular scale. So here's our friend, the phospholipid bilayer in DNA. There's an individual phospholipid, glucose, and that's about where we can wrap up our tour of scale. You can go and play on this website. You can also zoom way out and look at much, much larger things just for fun. Okay, so hopefully that tour of biological scales was interesting and peaceful and also maybe slightly relaxing. So just once again, so prokaryotic cells tend to be somewhere in this smaller size range. Eukaryotic cells tend to be in a larger size range, usually around 1 to 2 orders of magnitude larger. And so now this leads us to a question, which is, how is it that the eukaryotes have managed to get so much larger than the prokaryotes? And I think to fully appreciate this question, we have to think for a little bit about why it might be a problem to be bigger. And I'm going to highlight 2 problems with being big. And the first has to do with the cell's work surface. So how big its surface area is compared to how much volume it contains inside. And the second is a problem that's called the diffusion problem. So for problem number 1, let's walk through some calculations. So we're going to work with some cube-shaped cells because it's easiest to calculate volumes for cubes. So if we start out with a cube with lengths of sides of length 1, then the cube's total surface area will be 6, right? Because each side is going to be 1 squared times 6 sides gives you a total surface area of 6. And then meanwhile, the cube's volume is going to be 1 cubed because length times width times height is 1. So that's going to result in a surface area to volume ratio of 6/1 or 6. So now if we grow the cube so that each side is now length 5, if we do the same math for surface area and volume, the sides are each going to be 25 now times 6 sides, so total surface area is 150. And our volume is going to be 5 cubed, so it's going to be 125. So now the surface area to volume ratio has dropped down to 1.2. So what if instead of just getting bigger, the cell divided? So then instead of a big blobby 5 by 5 by 5 outcome, what we've now got are 125 cells, each of length 1. So the total surface area of those 125 new cells is going to be 750. The total volume is still going to be 125, and so now we're back to a surface area to volume ratio of 6. So it's hard to be big because you lose a lot of that working surface area. So now we're on to the second reason why it is hard to be big. And this reason is called the diffusion problem. And to give you a sense of this problem, here is an animation that's going to show you a single oxygen molecule bouncing around in the air. And what I want you to notice is that the random movements of the atoms or molecules, either in the air or in water, bumping into each other, means that any given molecule only winds up traveling very slowly across larger distances. And based on this understanding, biologists have done some rough calculations to estimate how long it would take for a substance, so in this case, I'm going to highlight a protein, to travel across the cell if that substance's movement only depends on diffusion. So here's an equation that can be used to calculate that. So here's time scale, and then the distance traversed, and a couple of other characteristics here as well. So just notice mostly that the time depends directly on the distance that that protein needs to travel. So if you start to crunch these numbers, what you come up with is that for an E. coli cell, it only takes about 10 milliseconds for a protein to travel 1 micrometer across that E. coli bacterial cell. So for an E. coli, that's no real problem. Now, these other two cell types here are eukaryotic cells, so in a size range from 20 micrometers up to 1,000 micrometers or a centimeter. And now we're running into some potential challenges. So it takes 10 seconds for something to get across a generic eukaryotic cell. And what that means is that trying to get anything done in that cell is at risk of taking forever on a cellular timescale. 10 seconds is a long time. And if you're a cell like a neuron that has a lot of long appendages, it can be quite long, just forget it. We do not have 20 days to react to our environment. We have got to be faster than that. So hopefully this highlights that there are some serious problems that can be tied to growing bigger. And so let's come back to our question here of how eukaryotic cells might have solved this problem to grow so much larger than prokaryotic cells. Because after all, we do observe cells of this larger size in the real world. So biologists' main hypothesis is that this is achieved through something that's called compartmentation. And I find the easiest way to think about compartmentation is to think about something like a house. Most of our modern American houses are divided up into different rooms. So here in this architectural drawing here, we've got a living room, we've got a dining room, we've got a kitchen, we've got a bedroom, we've got a bathroom. And so like the opposite of this might be something like an extreme open plan house, where the house itself is just one big room and all the activities in the house take place in that one big room. And I think it's useful to pause the video here for a moment to see if you can think of some advantages to compartmentation based on this house analogy, and then I'm going to go on to describe what biologists have generally come up with. You can also think about some disadvantages. So now here are some of the advantages that biologists have highlighted for compartmentation. So first off, if your cell is compartmentalized, what that means is that you can concentrate specific processes in specific locations. So if we use our house analogy over here, it's a lot easier to cook in a kitchen because you've got all of your ingredients right there. You can just pull the ingredients out and get to work. Second, compartmentation makes it possible to reduce that diffusion time problem that I just showed you. And we're going to learn how this works in a lot of greater detail over the next couple of lectures. There's going to be more on that soon. Collectively, these characteristics have allowed cells to become bigger. And an interesting thing happened at the same time. As cells started to get bigger, their genomes, which are the instructions for how to accomplish activities inside of the cell, also started to get bigger and more complex. And this, in turn, this increase in complexity allowed for the evolution of multicellularity. So then multiple cells that are often each specialized on different tasks, collaborating together. All right, so that all sounds great, but what about disadvantages? I'm actually going to leave you with a cliffhanger for a moment. There's at least one big disadvantage to this compartmentation, and we're going to see exactly how larger eukaryotic cells manage this bigger disadvantage later on in the semester. And in spite of this disadvantage, eukaryotic organisms are now very common across the planet. So now this is just for fun, but this is a graph from an amazing study that a group of biologists did, and they published in 2018. And what this graph shows is a picture of the total biomass of all living, and then also a couple of non-living things found on Earth. So here we've got our prokaryotes, so the bacteria and the archaea, so the orange and purple. And so the bacteria make up 70 gigatons and 7 gigatons, respectively. And then compared to that, here's plants. So plants are clearly the most overwhelmingly massive. So they make up 450 gigatons of all the living things on Earth. And then animals, kind of humble, are just down here in one corner at a puny 2 gigatons. So then if we enlarge the animals, things actually get kind of a little more humbling, because as you see here, the majority of animal mass is in the form of arthropods and fish, plus some things like some different types of worm-like creatures, so the nematodes and the annelids and the cnidarians. And it's also a little interesting, humbling, and a bit sad to notice where things are for humans, for livestock, then wild mammals, and wild birds. And at the moment, most of this is topics for future courses, but there are things I want you to be aware of in general. So from here, now let's turn our attention to a slightly different question, which is a question of how this compartmentation might have gotten started. So how is it possible to have gone from one of those simpler prokaryotes to a eukaryote? And in this case, there are actually two different hypotheses. So one hypothesis here is called the infolding hypothesis. And I want to show you a quick animation to help you understand this hypothesis, and I'll do a voiceover for it as we watch. Okay, so here we have a cell, identifying some structures in it. Here's a protein binding to another protein on the surface of the cell, and what you just saw there was part of the cell membrane pinching off onto the inside of the cell. You'll notice, so this little pinched off bubble of membrane is traveling, and now it is attaching to another piece of membrane that's inside of the cell, so it's able to release that protein that's bound to the outside, and the receptor protein on the inside of the cell is actually headed back out to the surface. And now some of those proteins have pinched off and they're headed off in a different direction. And then in this animation, so we see some of these bubbles headed back out to the cell membrane. Others are here waiting, full of some kind of exciting ingredient. Wait for it. Okay, so things being released from the cell. Now some kind of signal happens, and all of those bubbles fuse and release their contents outside of the cell. The idea here is that through this process of the bits of cell membrane getting pinched off of the outer cell membrane, it's possible that pieces of those cell membrane pinched off traveled inside of a cell and then stayed there. So that's the idea, that organelles, that's how they're formed. So how about our second hypothesis, then, this endosymbiont hypothesis? So here's how the endosymbiont hypothesis works. So first off, you have to know that one of the ways that some types of cells get energy and food is by swallowing other cells and digesting them. So here is an incredible image of that happening. This is a white blood cell shown here in the yellow. So it's a colorized scanning electron microscope image. And what this white blood cell is doing is it is swallowing an anthrax bacterial cell right here. You can see that tube disappearing into this white blood cell. You can actually see one that's been swallowed down here previously. So the idea behind the endosymbiont hypothesis is that maybe what's happened is one cell swallowed another, and then instead of killing and digesting the swallowed cell, what if it kept the swallowed cell instead? And what if that swallowed cell provided something useful to the cell that swallowed it? So that's the idea behind the endosymbiont hypothesis. Maybe the organelles found inside of a cell are ancient other cells that were swallowed and that were kept and passed on. So now, which out of those hypotheses do you suppose is correct? Is it the infolding hypothesis or the endosymbiont hypothesis? So historically, the infolding hypothesis was the more popular hypothesis. But that changed back around 1967 thanks to the work of the scientist that's pictured here, Lynn Margulis. And what Lynn Margulis did was she went around gathering together a whole bunch of evidence in support of the endosymbiont hypothesis. And I've listed a lot of that evidence here. We'll go through it in a second. But the thing is, prior to the point when Lynn Margulis did that, nobody had actually gone around and gathered all of this information and put it together. And when Lynn Margulis initially did this, the general scientific consensus, was that it was infolding, case closed. Not an interesting idea. And in fact, the infolding hypothesis was so generally accepted that it took Lynn Margulis 15 tries to get her very meticulous research findings published. And yet now, a lot of people tend to think that the endosymbiont hypothesis explains the origin of organelles in eukaryotic organisms, and that infolding has nothing to do with it. But I like to point out Margulis' story because it's a reminder that you should never give up. Keep on trying. Sometimes it may take you 15 tries, but sometimes those 15 tries are necessary. And in some ways, amusingly or ironically enough, now people tend to take the endosymbiont hypothesis for granted. And they don't think so much about the infolding hypothesis. So given all that, let's take a look through some of this evidence. So if we look specifically at two of the organelles we're going to learn about, the mitochondria and chloroplasts, here's some really compelling features that support the endosymbiont hypothesis. These two organelles are very similar in size to a group of the prokaryotes, the eubacteria. Mitochondria and chloroplasts also contain their own DNA, and the DNA that's in mitochondria and chloroplasts is strikingly similar to the DNA of bacteria. It is a single circular chromosome, and there's very few proteins present in these organelles. And that DNA that's found in the mitochondria and chloroplasts also replicates in a fashion that's similar to how bacterial chromosomes replicate. So we'll see more on that soon. Along with that, both the proteins, aka enzymes, and the ribosomes that are found inside of mitochondria and chloroplasts tend to look a lot like the proteins that are found in eukaryotes. And they share a lot of characteristics with the proteins that are and ribosomes that are found in bacteria. And lastly, mitochondria and chloroplasts can reproduce independently from how the cell reproduces overall. And again, we're going to see more about that coming up. Okay, so that all sounds pretty good, right, for the endosymbiont hypothesis. But there are some important caveats, and there's some important evidence that does not support the endosymbiont hypothesis. So in reality, in many respects, the jury's still out on what's the explanation for how eukaryotic organisms came about. So one big challenge is that the organelles, such as a mitochondria, are not completely autonomous, or in other words, not completely independent from the nucleus and the rest of the cell that they're found in. And so to show this, this is an illustration of a set of really important central proteins that are found in a mitochondria. And they're actually found in a membrane that's inside of the mitochondria. And this cartoon shows you the proteins that are used to produce energy in a cell. And the thing about these proteins is that some of the information that goes into the construction of those proteins does come from the mitochondria. So that's all these little protein subunits shown here in red, right? So these are individual amino acid chains, and then the collective is going to be what generates the protein. And then the rest of the information to build one of these proteins actually comes from the cell's nucleus. So this whole protein on the left here, it's an aggregation of information from the mitochondria and information from the nucleus. So, how can we explain that? It's definitely not the result of a simple endosymbiosis event. It's pretty complicated. And, we don't really have a great explanation for this. And then, along with that, it gets to be really hard to explain the origins of another cellular structure, the nuclear envelope, by endosymbiosis. And we're going to talk some more about why it's hard to explain the nuclear envelope soon. Okay, so to recap from this video, so in this video I've shown you that cells consist of two essential features: cell membrane, which surrounds genetic material, and that genetic material makes up the hereditary information passed from one generation of cells to the next. We've seen some of the key differences between prokaryotic and eukaryotic cells. We've also talked about the leading hypotheses for how eukaryotic cells are thought to have gotten their start. This should all set us up well for our next project, which is going to involve taking a tour of the different kinds of structures and organelles that are found within eukaryotic cells. I'm really looking forward to this exploration. It's going to help build out our picture of how cells work in multiple ways.